Hello, everybody. So in December, the AT Society, together with the AT Clinical Research Network, set up a Zoom call, or in fact, 2 Zoom calls, to discuss the status of clinical trials for AT. Some of you may already have seen a lay summary that we did for that. But what I want to do today is to try and relay to you the discussion that we had about those clinical trials. So first, let me just try to get the screen sharing working. Okay, so what I would like to do is try and help you understand the status of these trials, the scientific rationale for them. I'm going to go through a few slides at the beginning to try and tell you about the science, but I will try and keep that very simple, and the strengths and weaknesses of the trials. The goal here is to enable you to evaluate the trials. I cannot and will not recommend any particular trial, but to get you in a position where you can hopefully make some of the decisions that some of you may be faced with having to make. So first, I'm going to try and give you a little bit of the scientific rationale behind it. As I said, I will try and keep this simple. So I've told you previously, for those who attended the Science Simplified things, that in our cells, we have our DNA, which is code to make everything that we need in the cell, to make the cell function, to grow and divide and do its task. That DNA is built up of 4 building blocks, which are called ATCG, but we're not going into that. From that, a subcode, a smaller subcode is made, but I'm not going to talk about this in detail. This is called messenger RNA, has similar, although slightly distinct building blocks. And this code is used to make proteins. And proteins can be thought of the worker molecules of the cell, although sometimes they're structural molecules, but they provide the tasks, do the tasks that help to make the cell grow, divide, and do all its chemical reactions. And these are made up of 20 building blocks. So our DNA is organized in a way as a linear molecule with 2, and you can think of it as a ladder with 2 rungs. And these are these 4 building blocks organized in a sequence. And this ladder is then twisted to make a double helix. And I'm sure many of you have heard of the double helical structure of DNA. It's actually not entirely a single total linear molecule. It's divided into chromosomes. From that, the messenger RNA is a subcode that has just the information to make a single protein. Proteins are completely different in structure. They're tertiary in structure, so they're 3-dimensional, and it's this complex structure and shape and how they move that allowed proteins to do the tasks that they had to do. In terms of thinking about AT, so AT is Ataxia-Telangiectasia, the disorder. We have the ATM gene, and this stands for AT mutated, and it's called that even in a normal cell. It just means that it's the name of the gene that, when mutated, gives AT. So it's a part of your DNA. But then there's an AT messenger RNA that has specifically the information to make the ATM protein. So the root cause of AT is because of a change, and we call this change a mutation, in this sequence of these building blocks in the ATM gene, that bit of the DNA that has the code to make ATM protein. Sometimes one of these may be missing, sometimes they're changed, sometimes there are bigger deletions, loss of them, or sometimes even insertions. And what that means is that there may be reduced or ATM function, and ATM can't do its tasks so well. So a way to think of this, an easier way to think of this, is it's like an instruction booklet. For cells, it carries all the information to make these worker molecules, and the cell's worker molecules then are made from the instructions in the DNA. So the question that we need really to know, to be able to think about how do we overcome the fact that your children's cells are not making ATM, we need to know what ATM does. And it turns out that our DNA is quite a complex molecule, and it can be very damaged by other chemicals or by radiation, and in fact by our byproducts from our own cellular metabolism. And in fact, some processes also deliberately put in particularly breaks into the DNA, particularly immune development. Because your DNA carries the code to make this vital information for our cells, the damage must be repaired for the cells to function. If the proteins are damaged, and they are damaged, that's not a big disaster because you can make more because you've got that code. But if that actual code is damaged, then you can't go and make more protein. So it's really important that we do have a good process for repairing this damage, because it definitely happens just from our own metabolism. And it turns out that ATM regulates many processes that help cells repair and recover from DNA damage, and particularly from when both of these backbones of that ladder are damaged. So you can think of it as if the instruction booklet has damage or errors. The worker molecules, when they're made, are damaged and cannot function correctly. So you can think of a cell as having an infrastructure that is faulty. Now, so that's the main function of ATM, or when I say the main function, it's the function that we've known about for the longest. It has this role in helping the damage to our DNA get repaired. But there's another important component of the cell which I want to tell you about. That's called the mitochondria, which is relevant in the context of some of the clinical trials. So this is this kind of cross-section through a cell. This is the nucleus where our DNA is located. And what isn't the nucleus is called the cytoplasm. And in the cytoplasm is an organelle called the mitochondria. Most of our DNA is located in here. There's actually a bit in the mitochondria. And mitochondria are organelles then within the cytoplasm. And they're effectively the powerhouse of the cell. They provide the energy for the cell to do all its tasks. But they also emit what we call reactive oxygen species, which is this chemicals that can damage our DNA. And it turns out that there are two additional functions of ATM in addition to that function of repairing the DNA. So there's mounting evidence that ATM helps mitochondria to function properly. And there's evidence for what we call mitochondrial dysfunction, i.e. they're not functioning correctly in AT cells, in cells lacking the ATM protein. But also ATM regulates processes that counteract these reactive oxygen species that are emitted from the mitochondria. So in fact, often mitochondrial dysfunction results in increased ROS generation, but then also ATM helps counteract that ROS. So it's like a double whammy of impact. We don't know, and it's an important question, of how much this mitochondrial function might contribute to the progressive ataxia. But what is known is that there are some other disorders with mitochondrial dysfunction that do show ataxia. So there's I think there's really mounting evidence that the progressive ataxia of AT may be derived from multiple factors, and the impact of dysfunctional mitochondria could well be a contributing factor. So then the second question that I think is quite important to ask is, why do individuals get progressive ataxia? And it's this part of the brain that gets progressively lost. This is the cerebellum, and within the cerebellum is the Purkinje cell layer. And this literally shrinks as AT individuals get older. So why does that happen, and how do we overcome it? Well, the long and short is we don't actually know, but I'll tell you some of the possibilities. So just cast your mind back. I told you that this code is used to make a subcode, which is used to make protein. And this process here of making this subcode from your long linear molecule is called transcription. The second step is called translation, but I'm not going to talk about that. And neurons make a lot of proteins. That means they're very transcriptionally active because they're making a lot of these subcodes. They're translationally active as well, but this is the point of relevance for what I'm going to tell you about. And it seems like the whole act of doing transcription can cause some DNA damage. So I'll just tell you a little bit more about that. If this is all too much, just switch off for a couple of minutes. So when our DNA is not being used, we've got a huge amount of DNA, and to fit it all into a little cell, it has to be very tightly compacted. It's a bit like putting things that you're not using away in drawers. When it's not being used, it's tightly compacted and wrapped around and protected. Some cells are using some bits of your DNA, some are using other bits of your DNA, and some are not necessarily doing that much transcription. But neurons are very transcriptionally active, and that means they've got a more open configuration. Effectively, those drawers are slightly more open. That means that they're more exposed to those ROS and other DNA damaging agents. And that may be one cause of why neurons seem to be particularly sensitive to loss of ATM protein. But also the very active transcription can cause DNA damage. I might just go back because I don't think I explained it that well, but I told you that we lose this Purkinje cell layer in AT, but other cells function perfectly fine. They're not functioning actually perfectly, but certainly we're not losing them. So it's the question we're asking here is what is special about these Purkinje cells that make those rather than other cells get lost. Okay, so I've told you one thing is they're very transcriptionally active, but another aspect is this mitochondrial dysfunction, and that may affect the neurons more dramatically than other cells, partly because they're very active, they may be emitting more ROS, and then if there's dysfunction in ATM, that may result in even more ROS, and because ATM is not good at counteracting that. But there may also be less energy produced, because I told you these mitochondria are the powerhouse of the cell. So maybe we're not making enough energy to keep those neurons functioning. And that may be particularly important in neurons. Also, when there's more DNA damage, you can get more inflammation. And the inflammation itself can actually feedback and give you more damage. So it's kind of like a feedback pathway. And that could all result in the neurons dying. And I think it's important to know that there's a specific process in the cell that is deliberately removing non-functioning or damaged neurons. So maybe this is a particular area that's highly sensitive, maybe to the mitochondrial dysfunction, or just requires a lot of ATM protein to maintain its very active status. So that's going to be the end of the sort of basic science, you'll be pleased to hear. But now let's think about ways that we can try and use to treat AT. We could try to find ways to allow these Purkinje cell neurons to survive and function even though they haven't got ATM protein. So, for example, if inflammation is a problem, maybe we can reduce this with anti-inflammatory agents. Or maybe we can compensate in some way for mitochondrial dysfunction. And there are agents that seem able to do that. So they're more acting on ATM function. Another possibility is if we can change the mutation in the ATM gene to allow normal ATM protein to be made. I'm not going to talk very much about that, but at the end, I will tell you about an approach that's being used in that direction. Another very different approach is to try and give cells what we call ectopic, or that little code for ATM that now doesn't have mutations in it. If we can put that into a cell, then maybe the cell can function normally. That is a very valid approach, and there is lots of work going on in that area, but it's not yet at the stage of clinical trials, and that wasn't covered in our Zoom call. Sorry. Okay. So before I start talking about various clinical trials, I want to tell you about the type of clinical trials there are, because I think you'll hear these words and they might be a bit confusing if you don't know about them. So trials can be open label trials, and that means both the participants in the trial and the people carrying out the trial are aware of what drug treatment individuals being given. Another type of trial is called double-blinded, and that's where neither the organisers nor the individuals know which treatment is being given. This often will take place when there's a control placebo group and a treated group, or if there's two different drug uses are being used. And the rationale for that is to reduce operator bias. Then trials can be divided into three categories. There's phase 1 trials, and the aim here is just simply to find the highest dose of a new treatment that can be given safely. So it's not assessing whether the drug's working or not. You just want to know whether there's undue side effects. And that isn't needed if drugs have already gone through that for some other reason. And in fact, all the ones that we're going to talk about have been through phase 1. So there aren't any phase 1 trials being used on these 80 individuals. Phase 2 trials are aimed to assess the efficacy of the treatment. Everyone usually gets the same treatment, the same dose, or you may get a couple of different doses, and there may be a placebo group. It depends on how the trial is conducted. And then a phase 3 trial aims to compare that treatment with other treatments or with no treatment. And in this case, the participants, which one of those arms the participant goes into, may be picked at random, and that's called a randomized trial. Or it may be double-blinded, where nobody knows who's getting what until a later stage of the trial. Now, I'd like to just mention a point, really two cautions in over-assessing the results of the trials that you're going to hear about. It turns out that there can be improvements during the first 4 to 8 weeks just being due to being on a trial. Psychological, physiological effects. And this was discussed more in our second Zoom call. So improvements over the short term, you have to take with a little bit of caution. They're encouraging, and don't let's be, that we shouldn't dismiss them at all, but do be aware that there can be these sort of short-term improvements just by being on the trial. And the other thing is, when assessing ataxia particularly, 4 months, when some of the trials are only 4 months, it is short term, and those improvements or any worsening effects can be maybe reflective of slow progression, but maybe just due to other changes. Okay, so I'm now going to go through 3 trials, and the first trial is this A-test or Eritx trial. And this is this is the one that's been carried out for the longest and had the most number of patients. Blood is taken from the patients, and then the red blood cells are infused with dexamethasone, and then that blood is given back to the individual. So they've got their own blood infused with dexamethasone being given back to them. And this means that the dexamethasone is slowly released into the body, and it turns out this reduces side effects. Dexamethasone is a corticosteroid and a known anti-inflammatory agent. So it has the potential to reduce any problems caused by inflammation. And as I told you, there is some evidence that damaged neurons may even be removed. So it has the potential to stop that, although we don't know that. However, it's important to say that there is another very different explanation possible, and that's being examined during this ATES trial. And it's quite complicated, and I'm not going to discuss that other possibility. So I think it's important to realize that we don't know exactly how dexamethasone is functioning, although it is important to know it's a well-known anti-inflammatory agent. So this was a well-conducted trial, placebo-controlled, so there was a placebo group, and the individuals were blinded, so they didn't know if they were going to get the treatment or the placebo. And the idea of that is to reduce any of these effects that may be caused just by being on the trial. And then at the end, all the placebo-treated individuals could progress to having dexamethasone if they wanted. What's quite difficult in these trials is scoring for ataxia. There are a number of different scoring systems, of which SARA and ICARS are common ones. Because this was an international trial with different centres doing the analysis, it was important to have a standard scoring system, and that was done quite carefully, trying to make it as similar as possible between centres. And there were 175 patients recruited, and that's quite important because there are mathematical ways of assessing small changes to see if they're really significant. And the more patients you have, the better. And this was carefully worked out, working out how many patients you need to make the results statistically significant. So it was found that there were no side effects, no differences to the control group were evident. The results from this trial after 6 months were encouraging, and these have been published, and there was evidence of improved ataxia scores. And what was discussed in this Zoom meeting was the preliminary results from the 1-year analysis, and they looked similarly encouraging, particularly the 6 to 9-year-old participants who received a higher dose of dexamethasone. So actually individuals could receive 1 of 2 doses, and there seemed to be more improvement with the higher dose. So that's what we call a dose response effect, which is always encouraging. However, I cannot discuss these findings with you. I know you're all eager to know them, but I cannot discuss them in any more detail with you because they're being written up for publication. And I know that may seem, well, you're involved, you participated even, why can't you find the results? And it's important because these findings undergo what we call peer review. So they're carefully assessed and then enter into the literature. And you need to go through that process to be able to progress to another trial and for the results to be well recognized and evaluated. So the future plans are that the individuals in the trial will continue receiving the drug. That's what we call an open label extension. And there's also applications being made for further trials to the FDA and also European agencies, and possibly also just to try and allow access to the drug even without a trial ongoing. But you have to be aware that this is, it requires the use of hospitals to be able to take the blood and give the blood back again. Okay, so second trial that I'm going to talk about is by a company called IntraBio. And this talk was given to us by Taylor Fields from IntraBio. And it's about a drug called IB1001. Now, N-acetyl-D-L-leucine was a drug originally used to treat vertigo. It was known to be well tolerated and safe, so it had been through phase 1 trials, and it had some benefits for treating vertigo. The IntraBio have developed a derivative of this called IB1001, and the major advantage of this derivative is that it crosses the blood-brain barrier. So some of you may know that there's a sort of barrier by which many drugs will not enter into the brain. And obviously, if you want to treat the ataxia, it's really important we do get the drug to the brain. So this is a major improvement, a major advantage of this IB1001. And this has been currently used in a clinical trial for something called Niemann-Pick disease type C. And this disorder has progressive ataxia, dysarthria, dysphagia, and it had a positive clinical impact with improved symptoms and quality of life scores. There were also, there's a mouse model for this disorder called MPC1 mice. And in those mouse studies, so those mouse show what's called sigmoidal gait problems, and there was improvement in that. And that's basically their ability to walk in a straight line. What's important to appreciate, however, is that there are mice lacking AT, but they don't show progressive ataxia. And that's true of nearly all the DNA damage response disorders where the patients show progressive ataxia, the mouse models don't. So what's being monitored here is a bit unclear, and how it would relate to progressive ataxia in AT is also therefore unclear. So Niemann-Pick disease is what we call a lysosome storage disease. And lysosomes are the bodies in our cells that have worker molecules, and they break down and help to remove or recycle larger molecules and waste products. And IB1001 seems to restore that function. It seems to affect the membrane potential, that's the charge across the membrane. But all this lysosome function is very distinct to AT. There's no direct evidence that AT has any lysosome dysfunction, but we'll come back to where there might be an overlap. So IB1001 is currently being used in an open label rater blinded trial for AT. It's orally administered, and as I said, it's cross the blood-brain barrier, gets distributed to the major organs. And there have been 6 patients who have been assessed in a study in Germany, and they were using those SARA scores to assess ataxia. Of those 6 patients, all were assessed at 1 month post-treatment. 2 were assessed at 6 months, and 1 patient went on for a year. The scores at 1 month were improved in all patients. But remember, that's a very small timescale. And then there was continued improvement at 6 months in one of the patients, which plateaued at a year, but the other patient who had a low SARA score initially didn't improve over those 6 months. The mechanism of action is unclear. It's probably multimodal, and there was quite a lot of discussion about that at the Zoom call. It could potentially be affecting mitochondrial function, and for instance, could diminish ROS levels, or it could affect the membrane potential, which is very important in neurons. So basically, there is some positive signs, very short timescales, really, and a small number of patients. There is now an ongoing trial at Papworth, and there's one in the US and one in the
EU, and my recent understanding is that the one at Patwood is only for older individuals. I just mentioned this last point because though it was in the discussion mentioned whether it could be used in conjunction with dexamethasone. And the general feeling was there should be no kind of scientific reason why they couldn't both be used. And that sort of approach may be possible in the future, but I think there would probably have to be very careful monitoring if that was carried out. Okay, so the last trial involving several patients is on a compound called nicotinamide riboside. And nicotinamide riboside, I'm going to call it NR, is used to make something called NAD+. And this is another source of energy and a regulator of many cellular processes. There is evidence that it can become depleted in certain cells when there's excess damage. There are some reports of NAD+ depletion in AT mice, but that's not universally accepted. Some other studies don't find that. What's clear is that in one model system, which is not AT, there is evidence that it's the NAD+ depletion that is leading to the ataxia. But as I said, there's not really direct evidence yet of that in AT. NR, however, may also, there's evidence that it also improves mitochondrial function and may reduce loss levels. So the trial that I'll tell you about in a moment, there was a trial based in 80 mice where they were given NR, and they were reported to show improvements in neurological function and viability. But remember, 18 mice don't show the progressive ataxia. So whatever was being monitored here wasn't necessarily the same as the progressive ataxia of patients. I think it's important to realize, though, that NR is found in some fruits. It's a common dietary supplement. It can actually be purchased. So it's not necessarily anything that's going to be harmful to individuals. There've actually been two trials involving nicotinamide riboside, but one was in the Netherlands by Michael Williamson, and that's the one that was mainly discussed, but there's also one in Norway. So the one in the Netherlands was an open label proof of concept study involving 24 patients. There was no placebo control, but there were both classic AT and AT variants. Patients were treated for 4 months, then that was followed by 2 months when the treatment, they were still analysed for 2 months when they were stopped treatment. And the ataxia scores were scored using SARA and IPARS, and there was a dysarthria rating score as well. There was significant improvement at 4 months of treatment compared to the scores when they started, and the gains decreased during the 2 months washout, i.e. when they weren't receiving the drug, which is encouraging because it makes you think that it is to do with the treatment. That improvement was independent of age, AT phenotype, i.e. whether you classical variant, and included both kinetic function and speech subscales. I'm not going to go into the impact on the immunological deficit, but in some cases there was some improvement. Something very similar was also observed in the Norway trial that involved 10 patients, but that's going on for longer, and one patient's been on the drug for 22 months. Again, no side effects were observed, and there was a similar level of improvement, although not on the immunological scores. So I think that's all very encouraging, but still this is fairly early stages. The limitations with the mechanism of actions, as I discussed, is not entirely clear. Whether it's a consequence of NAD depletion and helping the NAD depletion is a possibility, but that's not clear, or it could work via mitochondrial function. And in these trials, there haven't been a placebo control, or at least
this trial, and it's quite short duration. So we need further studies to really assess the impact of long-term treatment. Oops. Now, at the very end, so they were the 3 clinical trials that we discussed in the first Zoom call. But at the very end, there was also discussion by Timothy Yu, who's doing another approach using what we call antisense oligos. And this is very different, where we're not trying to live without ATM, but also without having progressive ataxia. Here we're trying to make normal ATM protein be made. And antisense oligos, ASOs, are small, what we call oligonucleotides, little bits of RNA that can bind to the RNA and can alter the nature of that ATM messenger RNA. Remember, that's the code just for making the ATM gene. And for some patients, it can allow normal ATM to be made. However, it's only a small subset of patients that are amenable to this type of treatment. The nature of their mutation change allows them to be able to make normal ATM protein by using these little oligonucleotides. So in a way, so at the moment, this has been trialed using a single patient who had a suitable mutation. So in a way, this is a trial of N equals 1. But I think, as you'll hear, it's a little bit more encouraging that may be applicable to broader patients. But I think it's interesting for you to know exactly where this is at and what's happening. So ASO, short oligonucleotide, was developed, and that was called antipexin, which would, in this particular patient, allowed normal ATM to be made. It was tested using cells from the patient, and indeed normal ATM levels were observed. There was normal function in a variety of different endpoints, and that's all excellent. The difficulty then in treating the patient is that antipexin has to reach the brain. So the way they did this was to inject it into the cerebral fluid, that's spinal injections, and it requires, the treatment requires 6 injections per year. And this isn't trivial. I mean, it can have bad consequences if there's problems with it. The patient, who was age 2 when all this work started, commenced treatment in January 2020, so is now 4, and is doing well. But as all of you know who've got AT individuals, even at age 4, it's really hard to say that you've stopped the progression. So time is required to see whether there's really any impact. The drug certainly is well tolerated, but they can't really assess the efficacy in the cerebellum of whether ATM is actually being made in the cerebellum, because we can't take cells out of the cerebellum to try and look and see if ATM is being made. They're trying to gain biomarkers to see if we can assess if ATM function is restored, but that's still at an early stage and hasn't really been achieved. So it's all looking good, but there's no concrete evidence that it's working yet. However, what I think is interesting is that this Timothy Yu's group took a study of 235 patients, 80 patients, by what we call whole genome sequencing. And they found that 15% of those had a mutational change that they think is amenable to this type of therapy. It doesn't mean they can all use the antipexin. Each one has to have a different antisense oligo made for them. So it's going to be costly to develop a drug for each variant. And also interestingly, that there was a mutational change in AT variants in the UK that was considered to be suitable. But we still don't know, as I said, if function is restored in the cerebellum, whether it affects ataxia or how long it will last. And having 6 injections per year is difficult. It's a risky procedure. And remember, this patient being treated is age 2, so at a very young age, really prior to the development of the symptoms. So what we don't know is for patients, for say variants where the onset is later, if they were given the treatment later, would that actually benefit them? Or even individuals who have started to have some symptoms, would the treatment benefit them? And they're questions that we all need to answer. Okay, so I'm going to finish there with what discussed today. The next Zoom, so we have a second Zoom call with the clinicians, and this covered a clinical trial involving a drug called triheptanoin, which the trial is only just starting, but we went through the sort of rationale for the trial and everything. There was a discussion about a clinical trial with a drug called OMAV, which has not yet been used to treat AT, but is used for treating Friedreich's ataxia. But it was interesting to know how that work progressed. We talked about the design of trials for rare diseases, which is a very important area, and also biomarkers to assess the effectiveness of these clinical trials. Okay, so I'll finish there. If any of you have any questions, do feel free to email me or get in contact with the AT Society, and I'll do what I can to try and help you.